Okay, great.
Yeah, thanks, Nicholas. All right, so welcome back everyone to our workshop on real-time analysis of synchrotron light source and nanoscale research center data using AI ML for APS-U First Experiments. So we'll continue with our session now focusing on dynamical experiments and both the applications of AI ML as well as the scope of AI ML in applying to these kind of dynamical experiments for better information extraction and interpretation. So we have three speakers who will actually talk about different techniques. So the first speaker is Andy Barbour from Brookhaven National Lab, and she will tell us about enabling real-time AI-guided photon correlation spectroscopy at CSX. So over to you, Andy.
Awesome, thanks. Thanks for inviting me. I'm excited to talk about this work. So from the things that I'm sort of interested in and do have 4 different hats, and so today's talk is kind of at the intersection of all of these things. And I hope that you take home from this more generally just the challenges with automation. Often flexible experiments are difficult to automate, and for much of what we do, it requires a large amount of domain knowledge, especially related to machine learning, in order to try to automate these things. And additionally, maybe, increase partners and collaborators for automation, I think helps with achieve address the challenges in terms of the amount of work that needs to be done and the amount of more data we produce in terms of automation. And it's worth it to have a thought about what is the best data format, like the units that you want to use, and then also modularity, and not just make a large monolithic thing that is very difficult to address. I know that many of you don't need an explanation of what an X-ray speckle is, but I know that it's a very broad cross-section of people here today. Basically, we can use coherent X-rays that are monochromatic, that produce a speckle pattern, because the X-rays are in phase, much like a laser, as opposed to something like an LED. And then if we collect a bunch of these images over time and monitor how a collection of pixels change their intensity over the course of time, we can extract dynamic information related to the sample at hand. Additionally, you can have different sort of geometries, and this is a reflection geometry of a very nicely prepared surface. And essentially, when you have the step terrace in the footprint of the beam, you get 2 speckles. So you imagine how this would make something like XPCS really difficult, since it's a statistic-based technique. So in 1991, that's when coherent X-rays were first used to observe a coherent diffraction pattern, and it took another 4 years in order to generate a paper to quantify the dynamics of that particular sample. And I think that's still very much the case today, in that we have a series of images. We need a lot of different tools before we get a final answer. And it's not uncommon for it to take 6 months to a year after a user finishes their last experiment to have a publication. And so at CSX, we're a soft X-ray beamline. I know the APS is going to be tuned better for hard X-rays. But this was sort of the first experiment that we did when the beamline was brand new, about in 2015. And it took, a fair amount of beam time to get this result, even though everything was static as a function of temperature. And we ended up doing a different kind of experiment. So already the XPCS that was envisioned was not the thing that actually happened. So this calls for a need to have flexible tools for, especially when you're developing beamlines that have more flux, where people are going to want to measure a lot of different things that haven't been measured before. Okay. And then the last thing I would say is I think it was, sort of interesting that the contrast that we had for this LBCO sample was the same as the sample that was published as one of the first XPCS experiments where they extracted dynamics. So in terms of bottlenecks for scientific productivity, soft X-ray detector, like the one used at CSX, requires a lot of data just to normalize properly. In addition to, just doing the analysis and repeating the experiments. We've even had, expert user groups come back 2 or 3 times just to make sure that what they measured was real. Additionally, the instrumentation is a challenge. The detectors for soft X-rays are noisy. XPCS itself is very sensitive to instabilities in the optics or the sample, since we can't directly know what's happening in real space. It's often, left to the user to figure out whether or not their dynamics are actually real or a consequence of instrumental instabilities. And additionally, at CSX, we service all of these diffraction geometries, and you can see just the variety of work that we do. And finally, new users that have never used coherent X-rays and don't come from these groups with 10, 20 years of experience using XPCS, and the computational resources for modern XPCS experiments far exceed laptop capabilities or even standard PC capabilities. So to deal with a lot of this large data, we took a page from CHX, where they've developed this paper mill pipeline to iteratively execute Jupyter notebooks and generate custom files in an automated way. But before we could even think about doing that for XPCS, we had to figure out how to reduce the images. And so some effort was spent doing that in an automatic way using data brokers search features to find the appropriate dark images and some other metadata that would be recorded along with the corrected and fully normalized detector images that would be the input for any XPCS analysis. In addition, this is nice because you can separate notebooks for jobs or purposes. So we have a separate notebook for XPCS, and this makes the notebook easier to read and review. And as we implement more and more machine learning, we can have a few notebooks run simultaneously in order to understand what the best path forward is. But not all scans are XPCS scans at CSX, and so we can use the metadata associated with each scan in order to make sure that we process data correctly, and using sort of just standard tools that are available. So I guess the big question is to automate or not to automate, and I think most everybody here would say yes, but there are concerns that I've heard from colleagues. So first of all, you're lacking sort of the context of the experiment, and that's the bringing the human in the loop part. There's also errors in the parameterization prior to computation. That could be from image normalization to the output of the correlation functions, or even fitting the functions themselves. And finally, there's the inappropriate extraction of quantitative results. And so I guess once you automate everything, how do we, as a beamline scientist or data scientist at the facility, address the temptations to use the automatic results as a black box? And here you can see this is a 2-time example that looks a little odd, especially once I tell you that it was supposed to be a static sample. So this line should be completely flat. I shouldn't see any decorrelation in the signal. So we have more photons, more problems. I don't need to explain any of this to anybody here. I think the big problem is that the time and people can't address the amount of data, and so we have to automate, but maybe selectively automating the things that give the biggest bang for the buck, which may be sometimes the more boring things to work on scientifically. But I think that supports future endeavors to do really advanced stuff with machine learning. Also, X-rays are a finite source, so more photons doesn't equal more science. So if we automate as much as possible, then we give time for people to explore the data in new and interesting ways. And finally, machine learning and artificial intelligence can help draw a focus to the most important things. And this is what I think is the first place to start in terms of selective automation, sort of what is bad data and what are good results. And I think just from my experience, it's okay to go backwards as you develop these things. Often you can't plan for everything. And I think that the best thing to do is once you know you need to make a change and go backwards, you do it in a concerted way so you have a clear mark in time. So we got some funding to quantify dynamics with potential artifacts and remove artifacts. But we're going to do it for both the CSX and CHX beamlines. And these beamlines, I think, are pretty different in their mission and their operation. CHX is heavily focused on XPCS, especially for SACs and GSACs, whereas CSX mostly works in reflection geometry. The camera technology for X-ray detection is very different for soft versus hard X-rays. And the duration of the experiments are also challenging. And then again, just the noise level for a single photon detector, precisely know where the photon is, and that's not currently possible with soft X-rays. So that leaves us with one option of using the raw time series of these coherent scattering images as the object or the input data in order to try to understand dynamics and artifacts and quantify those. Or we can just immediately switch to the two-time, which is interesting because it's doesn't have sort of the information that it's a common linking point. And so that's what we chose to do. So by using the two-time, you're no longer worried about sort of the quantum efficiency of the detector or the pixel size or the speckle size on any, dead pixels on the detector or anything like that, or the geometry. And the two-time is also attractive because you need less varied data. It's just an image, so who cares what the units of time is? And in this case, it's frames. It's the number of data points that we collected during the time series. It's also easy to alter the noise level, not by manipulating the two-time, but selecting the different pixel bins for the noise level, and to alter the lag times in the output image itself. And finally, you just need to have a really good normalization schemes for your input data prior to training your models. But the two-time isn't the only quantity that meets our needs for generalizability, because the two-time calculation is quite expensive. So Tatiana was the postdoc that worked on this tool. And we can sort of look at it from the standpoint of statistical quantities. In terms of XPCS, I think people typically like to look at the average intensity. And in this case, the result from this user experiment, there was a lot of wiggles in the autocorrelation function, which is sort of reminiscent of Paseo-like flow. But this was a solid material, and that kind of would be, a nature level paper, but more likely it was a problem with the instrumentation. And sure enough, that's where we found the problem. It was in the cooling water for the optics. And so we noticed that the center of mass of this peak was sort of correlated with the pressure of the cooling water. And simply by just culling the data and using sort of apples versus apples, sort of points in similar time, we were able to get a result that sort of made more sense. And so taking that sort of inspiration, we can engineer, we engineered 6 features, one being sort of just the integrated intensity, obviously the center of mass and the standard deviation were also chosen. And we were able to use these features to train a semi-supervised model using sort of 3 out-of-the-box architectures in scikit-learn. And this is a sort of this local outlier detection, elliptical envelope, and isolation forest. And so it's semi-supervised because the experiment, the model has only seen good data, not bad data. And in this case, we were able to get a pretty decent recall rate and not a false discovery rate. And you can tune this a little bit during the training process to get more error on the side of anomaly detection or false discovery. And finally, I think this sort of thing is not very heavy computationally, and it can be integrated in things with Blue Sky Q server or other methods. One of the sort of questions about this was this applicability with other things that don't have anything to do with XVCS, and sort of using these 3 models out of the box. And one might imagine the step scan tachography or CDI, where maybe you do have a speckle signal and you need to collect many images over time. You want to make sure that it's easy to add those images together. And this seems like a really efficient way to do that. Additionally, it is sensitive to sort of intensity changing and beam dumps. So you can use that to filter data that you should process or not process. One tutorial is sort of standalone tutorial to show maybe how useful it might be on other things with other detectors and other techniques, like diffuse scattering peaks or even narrow Bragg peaks. Seems to work fairly well. You just have to consider all 3 models together. And if they all 3 predict an anomaly, then it's definitely worth your time. And that's without retraining the models for this particular experiment, which is not one that anybody would want to spend any time doing anyway. And it's very different from an X-ray experiment. The second tool Tatiana worked on was indeed sort of this 2-time input for machine learning models. And so where we wanted to start was this sort of denoising the 2-time using a neural network and this encoder decoder, which is used for natural language processing and image problems. So it's pretty, I guess, well understood in machine learning academia. And here is an example of an equilibrium 2-time where you can extract the dynamical information normal to this line. But sort of putting this whole thing into a model and trying to get it to act correctly is challenging. It's a lot of memory. And so what we did instead was to chop these into 50 and also 100 frame sizes. And this gives us the ability to test many times with a lot of varied data. And you can see that the input would be something like this. And we told the model in the training process that the target or the answer should be something like that. And here you can see some preliminary results where this bottom graph is just the average along the diagonal. To extract the dynamical information, you would just fit an exponential function to this. and you would understand the dynamics. One question may be, how does this compare to sort of out of the box methods? PCA is actually very inaccurate. It artificially made the dynamics faster with potentially some other stuff out here. Other methods enhanced the noise and didn't really provide data that was even analyzable at that point. And sort of, if you're not familiar, the convolutional network architecture looks something like this, where you have your input, and then the encoder layer is something done by the algorithm to interpret the data, and then it compresses it in this latent space, and then it's decoded on the other end and returns, the output that you've trained it to return. I think non-equilibrium dynamics are sort of where it's at now with XPCS in terms of hard X-rays. I think most people are very interested in these kinds of systems. They're still quite rare in condensed matter. But the largest challenge to these kinds of data sets is how to extract meaningful information to then fit an exponential function and extract these dynamics. And so many a graduate student and postdoc have spent a lot of time playing with the size of a bin that would be integrated to then represent the dynamics at this point in time over the course of all of these. And so you can imagine that this is quite challenging. But fortunately, we were able to use much of what we learned from the first model on equilibrium dynamics. We had to alter how the normalization of the inputs were done. That was really important. And we had to tune differently the model itself, and all those details can be found within this paper here. And I would say the paper also references the GitHub link that has the trained models. So how do these things work together, and how do they perform? This is sort of the next step in integrating these things into a data analysis pipeline at CSX. So in terms of non-equilibrium dynamics, you can see it's much simpler to sort of bend. We actually get single line cuts, have more or less the same fitting error after these two times have been denoised. These are raw. And then the result would look something like this. So this already looks really promising in terms of understanding more about your samples and using every ounce of the data without having to spend more time collecting data. Another part is just sort of the fitting. So much of the automated fitting now fits the curve, it fits the whole curve or all of the frames. And it can be very difficult to try to automate that in a way that you always get the right answer. And it requires a lot of human eyeballs to look at the fits and make sure that they're reasonable answers. And so this is an example of sort of trust regions where we constrain the fit to frames based on the degree of correlation amongst the four parameters, and also from analysis of the residual of the denoised output from the encoder-decoder model. There was some question about whether or not you couldn't fit these denoised models with the global fit instead of fitting every single slice one by one. And here's an example of the the raw minus the denoise versus the raw minus the global fit. They don't look very different, but if you look at the difference of the difference, there are differences. It might be easier to see in terms of the linearization of this gamma parameter. And you can see that the raw versus sort of the global fit have two very different slopes, whereas our results with this that are represented by the red curve indicate that there could be a variation in the rate of change of the dynamics. So it's already a much more complicated non-equilibrium dynamic that we were able to extract quantitative information. We also get sort of outlier detection for free. So earlier I talked about this model with the outlier detection and it being able to predict an anomaly or not from different data sets. In this case, they should all be static. The 2 times shows a result that looks like this. And you can use out-of-the-box scikit-learn unsupervised to sort of group these so that you can analyze them appropriately, much like a human would. And then you can see for data set 2, these are the 3 results, which on their own, neither one of them are very useful. Instead, we can use the latent space from the encoder model. So you don't even need to let the entire pipeline run. You can just extract the information after you've encoded and compressed it. And you can see that data set 2 is very close to data set 1. And any human looking at this that knows something about XPCS would say, well, okay, they're both static, so nothing is different about these 2 measurements. Let's just move on to the next temperature, especially if this data set were expensive in time to collect, like if it took, half an hour.
Andy, 1 more minute, yeah.
Okay, thanks. So what's next is fully convolutional denoising. And classifying different kinds of dynamics. And we're working on an open library for all of the models that I presented today in one place for any user to use. And I think most of this is probably well understood. Things are going to get harder before they're easier. The interfaces, I think, are really important in terms of how to move forward. And with that, I'd like to thank all of the collaborators.
All right, thank you so much, Andy. Questions for Andy? Please unmute yourself and ask, or you can also drop it in chat. I have a question. So are there efforts to actually combine with like physical models for both of these equilibrium and non-equilibrium dynamics that you're actually looking at?
There is an effort by Oleg Chubar and Lutz Wiegard to do a lot of simulations on soft matter materials using lamps. And so we can imagine going in that direction. I think that's a possibility, but yeah.
And any comment on the spatiotemporal challenges, like, what kind of dynamics can be currently studied, like, what could be done in the future?
Yeah, so that's we've got this model already, this fully convolutional model, and that's nice because it doesn't mean that you need to downsample your 2 time. It can be of any size. And we're tuning it to deal with novel non-equilibrium dynamics, things that have like speckle echoes or bimodal dynamics. And so I think hopefully within another 3 months, we'll be able to publish that work.
Awesome. All right. Any other question for Andy? All right. If not, let's actually thank Andy for a wonderful talk. Thank you so much, Andy.
I'm having trouble stopping sharing. Okay.
Our next speaker is James Horvath from Argonne National Lab. So Jay, if you can actually start sharing He's going to talk to us more about using AI/ML plus XPCS for exploring non-equilibrium dynamics. And so over to you, Jay.
Thanks. Do you guys see my full screen presentation?
We see it in a presentation mode. So if
Okay. How about now?
it's great.
Good? All
Yep,
right.
thank you.
So thanks for the introduction. My name is Jay Horwath. I'm a postdoc at the Computational X-ray Science Group here at the APS. I'm really happy to be here talking to you all today, and especially I'm happy to follow up the previous presentation where we're actually going to be talking about similar concepts just from a different perspective. So I'm going to be diving into one sort of case study that we use where we're looking at non-equilibrium relaxation dynamics in a complex fluid as sort of a case study for how we can build XPCS tools for the future, especially after the APS upgrade. So I'm going to spend a little bit of time going through some introduction about the technique itself and the sort of problems that we face before showing my research on the topic. So as we learned before, X-ray photon correlation spectroscopy just starts with coherent sacs, right? We have incoming coherent X-rays scattering off of a sample, and then we collect a scattering pattern in just a normal fashion. Here we do a transmission, and we're looking at soft matter, so we use small angle X-ray scattering. But the key here is really that the coherence of the beam creates speckles in the scattering pattern, which we can actually use to track the dynamics. And so just to show a little bit how this happens, you know, we collect these scattering patterns in a time-resolved fashion. We can pick one region of the scattering pattern to correlate over time. And something that I'll come back to later is that we can perform these correlations all over the scattering patterns so that we can get you know, different dynamics in different directions, for example. But basically, we perform these correlations of the scattering pattern. And the way that I like to think of it is we first build it up into this two-time correlation, or C2, where we're looking at each pixel as the correlation between two frames of scattering data. Along the diagonal, we're correlating one frame with itself, so we have a very high correlation intensity. And as you move off the diagonal, the intensity decreases because, you know, we can think of it intuitively. The longer time delay you have, the less similar scattering or dynamic data is going to be. And so the traditional way to look at this type of data is actually not to use the two-time correlation function at all. Instead, we look at constant delay times, assuming that we're in steady state, where each delay time is this delta t, or the x-axis over here. We average along these parallel diagonal lines, reduce it to a one-dimensional plot, and then you can fit it to some kind of analytical or empirical model to get something such as a time scale or a velocity out. And so this works great for systems that are in equilibrium, because if you look at this two-time pattern and you follow one of these diagonal lines across the whole length of the experiment, we more or less have the same correlation intensity. So we're able to really reliably reduce into this one-dimensional state that's a lot easier to deal with. The problem comes up when we get to something that's out of equilibrium. So this is one example of such a system where if you follow this diagonal line again, you can see that the correlation intensity changes drastically. The behavior at early times is different than the behavior at late times. And so if we were to sort of decompose this two-time correlation into a one-dimensional pattern, we would lose a ton of information. And actually the assumptions behind the analysis aren't valid anymore, so you can't trust your results. There has been a lot of work in XPCS to sort of overcome this limitation for non-equilibrium systems. Here I'm showing two different, you know, what you might call coordinate systems of how you can sort of, instead of averaging over these diagonal lines, you can track the dynamics as a function of observation time on the diagonal axis and delay just with respect to a single time here, or in this rightmost plot, basically you're doing the same sort of scheme, just instead of taking a horizontal line cut at a specific time, you're actually using this delay time coordinate system like in normal XPCS analysis. And basically the point is that that gives you these sort of data where you can see the trend. You know, for example, we can see that as we move from light blue to pink, the relaxation time or the characteristic time scale of the dynamics steadily increases, and it's something that we can understand. And that's for this example of a non-equilibrium two-time correlation. But the problem is that sometimes you get something that looks like this, where we really don't know what to make of it at first pass. There's no clear trend in the dynamics. The observations are drastically different depending on what point of the experiment you look at. And even compared to a sample like this, where we might be able to say the dynamics are steadily slowing down through the course of the experiment, there's not really much qualitative interpretation that we can give to such a complex pattern. And the problem is that this type of non-equilibrium, severely transient and intermittent dynamics is typical of relaxation in complex or non-Newtonian fluids, which is our research area here. So now with this sort of motivation of our challenges with XPCS, I want to go into a little bit about the materials in the experiment. So just for a little bit of background, non-Newtonian fluids, in this case, we're talking about a colloidal suspension. That's what I'm showing here, where we have a dense, really glassy phase colloidal suspension inside of a rheometer, where basically you're twisting and applying shear. The idea is to measure the mechanical response or the viscoelastic properties. And what makes it non-Newtonian is that the viscosity changes depending on the rate at which you apply the shear. So for example, at low shear rates, we actually see decreasing viscosity. And then all of a sudden there's a critical stage at which we see a discontinuous shear thickening. And so these behavior are poorly understood. We have some phenomenological models that kind of describe it, but we want to look more from like a microstructural perspective with the goal of understanding how do microscopic dynamics impact the bulk measurable properties. And so we do this with XPCS. This is the same idea. We're using coherent X-ray scattering instead of having some generic sample. We're scattering through an X-ray transparent rheometer so that we can actually track the dynamics and the structure, because we get sacks at the same time while the material is under shear. And so to give a quick overview of exactly how this works, we do our standard rheological protocol. We start shearing, we increase the shear rate to see how the viscosity changes and pass the shear thickening transition, all while collecting structural sacks data. We hold it at this high shear rate to get some idea of basically how the material reacts under stress. And then basically, we all of a sudden set the shear rate to 0 and watch the material relax. And this is when we can do XPCS. And so just to kind of schematically show you, the idea is to figure out how does this complex two-time correlation relate to these bulk measurable properties where we are kind of condensing everything into a single measurement. And, you know, our hope is that somehow we can tie the dynamics to non-monotonic features, in this case, the shear stress, but just general mechanical behavior. Now, an additional problem with all of this is that there's a huge variety in the behavior of the dynamics. So if we had one sort of complex pattern, then we might be able to figure out a scheme where, you know, to help us understand what it's actually showing. But we have this huge variety. There's not really any clear trends. So what we want to do is figure out how can we use AI or machine learning to sort of categorize or classify these dynamics without human input, just to give us a little bit something to learn. And so I want to spend a little bit of time before I get into the details of what we do, going into some very basics of machine learning, just to get us all on the same page. In X-ray science and material science, we see a lot of AI applied for optimization where we have difficult or expensive experiments, and we try to figure out how to make them easier. We can do data reduction, where, for example, we collect too much data and we have to figure out how can we reduce it to a usable amount. And then in this case, we have the example of using AI to understand really complex phenomena where we don't have a physical description. So let's figure out what can AI pull out. In a lot of cases, you'd see the use of supervised learning, where we have results from previous experiments. We know what the right answer to a question we're trying to ask is, and we try to train a model for regression or classification to sort of perform the analysis for ourselves. So that's here what I'm showing, where we have pre-labeled data. I gave you the classes, red or blue, and what we're trying to learn is this line that separates the classes. So in your classic example, this could be separating cats and dogs, right? Feature 1 could be weight, feature 2 could be height. Cats are generally smaller, so maybe red is here. Dogs are generally bigger, and we're learning this line so that next time, if I just tell you height and weight, you can tell me whether you think it's a dog or a cat. In the unsupervised learning case, we do not have any labeled data, so classification becomes difficult. Instead, what we're looking is to distill similarities between features of the data that you provide, so that we can basically reduce it into simpler cases or into clusters. So here we don't know specifically where the line separating cats and dogs is, but we can recognize we have a cluster of samples with similar behavior over here, and then another one over here. And then that's where the scientist kind of comes in and says, what do these two things mean? So this is very broad. There's a million machine learning models that can do this type of analysis. We're looking at deep learning, which usually uses neural networks. So I'm going to go into a little bit about that. So neural networks are universal approximators, which is great for scientific data, because if we don't know what the function that describes the data is, then we might be able to learn it here. And the way that this works is you give input features. This is, you can choose anything about your data that you would think is important. And basically what we're learning is the weights in these connections between nodes. And so basically what's happening is you give your input, you have some not arbitrarily designed for a specific case, neural network architecture. You compare the output from the untrained network to the correct answer. And then basically what you do is train the weights connecting all of these nodes, and nonlinearities in the neural network is what allows us to actually learn complex data, assuming that you have enough training data. For image data or something like this 2-time, we use convolutional neural networks. So the idea is that instead of having these weights that connect to specific nodes, your nodes are these individual blue pixels. So we have sliding windows that go across your input data, and the goal is to retain spatial information. And this is basically how we can apply deep learning to image data in this case. So now this is kind of where we can pick up the actual research on XPCS data. Here we use an unsupervised autoencoder. So what that means is we don't have to have any ground truth, which is exactly what we need, because we don't know how to describe the dynamics. So we don't want to have to build in any of our biased assumptions. The way that this works is we use a convolutional neural network, like I described earlier. We have 2 stages, an encoder and a decoder, where in the first stage, we input raw 2-time correlation data. The neural network compresses it into this feature-rich latent space, which has a smaller dimension than the input data. And then the decoder model learns to decode it back into 2-dimensional space. And the way that we train the model is by trying to optimize the output. We try to basically optimize it so the output is exactly the same as the input. And so what we're trying to do here is basically make this latent space as rich in information as possible, so that now, instead of having to look at full image dimension data, we can take this reduced space and do some more simpler statistical analysis to try to understand what are we actually learning here. And so to just show this in a different way, we pass each 2-time correlation through this neural network. Each one provides a unique latent vector that we can then project into a reduced space. So now if our input XPCS data is 256 by 256 pixels, which is what we use, but it's important to note is a very small amount of data for these types of experiments. Instead of having 256 squared dimensions to describe the data, now our trained neural network is able to represent it in 64. And then, we can further use other algorithms to project it into this 2-dimensional representation, where now each point represents a unique data set, a unique 2-time correlation, and we can start to dig in and learn about the relationships between these data. So the first thing that we can do is to use clustering, like I mentioned before, to note regions of similarity sort of in this latent space, which maps the dynamics that we see in one experiment. That's what I'm showing here. Each color corresponds to a specific cluster label. You can see where they sort of overlap, and what that's showing here is that we see kind of a steady transition in dynamics rather than distinct clusters. But if we sample from each of these clusters, we actually see that they capture the change in dynamics. In class 0, we see very bright, broad correlation decays, and it starts to basically capture the increase in the speed of dynamics as we move to class 4. And so all of this is just to say it's a proof of concept, and that we are able to actually teach the neural network physics so that then hopefully we can use it to understand the complex data. One way that we might want to do this is to take sort of anomalous data that we might not understand and see if there's any similar examples. So that's what I'm going to be showing here. I selected these 2 sample 2 times because they show similar patterns. They show these fringes. For reference, we do understand where these fringes come from, but it's still a feature in the data that we would want to recognize, see how frequently it happens. How does the change in the frequency of the fringes occur? Something like that. And so then what we can actually do to learn about this is build neighborhoods in the latent space. So we can take our target images that I showed on the previous slide. We have a blue one and a green one. The position of the target in the latent space is shown by the open circle. And then we can just grab the nearest neighbors. And that's what I'm showing here, the 4 nearest neighbors for each of them. And what we can see is that they're actually very similar. So this is something where, in this data set that I'm showing you here, we only have 900 unique 2-time correlations. But if you're looking over an entire beam time, for example, you're going to have a lot more than that. So if we have thousands of data points and we just want to know, how frequent is this strange pattern that I saw? Now we can do this. We can sort of identify a neighborhood, tell the model how far out we want to look for similarity, and then base our, first pass assessment of the data on that. The second thing that we want to do is see, can we actually relate the microstructure to the bulk properties, like I said, which is kind of the holy grail in this case. So as you may recall from when I showed before, one of the things that we see is when we turn off the shear and we watch XPCS, we actually see these non-monotonic changes in the shear stress and the relaxation. So there's something happening. The dynamics are changing, but can we capture this? So what I'm showing here is basically the percentage of different dynamic classes at different times. You may remember that I said at the first part, when we're doing XPCS, we calculate these correlations over the specific regions of the scattering pattern. Well, so we can calculate the correlations at many different regions. In this case, we have 18 different XPCS 2-time correlations for each time step. We classify each of those 18, and what I'm showing you here is a bar chart, basically, of how frequent each of the dynamic classes is. We have purple or blue, which is slow dynamics, yellow or light green, which is fast dynamics, and basically what I'm trying to show here is that in these regions where we have minima in the shear stress and then increasing, we actually see slowing down of dynamics. So the point of this is that by just doing this wholly unsupervised method, we're able to pull out a little bit of the physics. We're able to tie the dynamics in these incredibly complicated two-time correlations to what we actually measure with the bulk properties. So this is sort of the end of the research before I get into what we're working on in the future. But just to sum up, what can we learn from AI in this case? Well, the idea is that by using unsupervised machine learning, we're able to actually distill a lot of information from really complicated data sets. We're able to tie this to the physics, meaning just kind of the average dynamics of the case. And then I showed two examples of how we could do this to sort of navigate a complex data set where we can identify patterns of interest or neighborhoods, and we can try to tie the microscale dynamics to the bulk rheology. But then the big question is, how do we put this to work? It's great in this one case where we have AI working for a specific topic, but what about at the beamline? This is something that everybody would need to use, especially as we have more and more data coming in, which takes more and more time to analyze. So first thing that I'm going to show you is that we've been working on implementing a web app, which would be able to use at the beamline, so that you can kind of take the machine learning knowledge requirement out of it and give users the ability to just sort of perform this first pass qualitative analysis of your data set while you're collecting it. And then the second thing that I want to talk about that we're working towards is tracking these dynamics in real time, so that when we have these complicated patterns, maybe we can detect it and optimize the data collection so that we make sure we don't miss anything, for example. So this, I'm going to kind of talk you through a demonstration of what we're doing here. Basically, I'm trying to set this web app up so that we can perform this AI analysis in essentially as drag and drop a fashion as possible. So there's a little bit of delay. I'm still working out some details exactly how this works, but basically you can upload your XPCS data set directly off the computer wherever you're collecting the data. We have it set up so that there's a pre-trained Rayo XPCS autoencoder in here. What I'm showing is clicking around different images in the data set to basically visually evaluate whether or not the model does a good job representing the data. In this case, we're seeing a lot of examples where it gets pretty close. And so process all images means run everything in the data set through the neural network and save the latent representation. So here we have 900 images, and I'm running this just on my laptop. It takes 10 seconds, but ideally, we'd be able to accelerate that a lot. One of the things the previous speaker mentioned is that we want to be able to have a lot of flexibility in the way that we analyze. Otherwise, we lose a lot of the information. And so I'm trying to make these tools as flexible as possible so that we can use different types of clustering algorithms, different types of methods for representing the data. We're seeing a little bit of a slowdown here, but the point is that we can perform this analysis like I presented right in real time. We have clustering, we can sample the different clusters, you can change the number of clusters on the fly. And so I'm just going to try to skip ahead here a little bit. And then what you can also do is sort of recognize, what did the different neighborhoods show? Here we're highlighting one sample that sort of looks like an outlier. And we see it's actually, really fast dynamics where we don't actually see much evolution, which is distinctly different from the rest of the data set where we can actually track the dynamics. So all I'm trying to show here is that AI is only as good as the usability. So here we're trying to build in a way to actually let users apply these tools while they're collecting the data. And so finally, we also want to be able to do this to be able to classify the dynamics at finer time scales and in real time. So the idea is we have these experiments, we have a continuous stream of scattering data coming. We can send this to a workstation to perform XPCS on sort of small and continuous batches and build up a dynamic two-time that looks something like this, where we're tracking data coming in. Hopefully we can capture changes, which is what I'm showing here. And then the idea is that if we have a good understanding of what dynamics look like, then hopefully we can build a good understanding of what transitions between different types of dynamics looks like, and be able to capture these as they occur. And so the benefit to this would be if you have slow dynamics and then all of a sudden you see something happen very quickly, then maybe you can change the frame rate of the camera, for example, so that you don't miss any information. Or even for the simplest case of this data takes a long time to go through after you collect it. So if we can at least understand where the transitions occur ahead of time, then it kind of, eases the analysis once you, sit down at the computer. Excuse me. And so the final thing is just a conclusion here. I want to go through everything that I talked about. And the big takeaways are that we can use this unsupervised machine learning to try to understand this really complex data without having to build in our human bias, which is especially important since we don't know much about the dynamics and especially the microstructure of these non-Newtonian fluids. We can use latent space clustering to build initial classification of the different types of dynamics, which allows us to track the dynamics with respect to bulk properties, and also to identify neighborhoods where we see sort of anomalous behavior. And then finally, the main point of this is that hopefully by incorporating AI at the beamline, we'll be able to accelerate science and be able to fully utilize the experimental capabilities that we'll have after the APS-U. So thank you for your attention.
Well, thank you so much, Jay, for this wonderful talk. Questions for Jay? Please feel free to unmute yourself or drop your question in the chat. So Jay, are you already working with like simulated data from the coarse grain simulations? And can you comment on the advantages of actually maybe training or pre-training it on synthetic data? And then before you actually kind of move towards the real-time interpretation in experiments?
Sure. So at this point, I'm still just getting started with combining the simulations effort here. But the benefit is that if we can have some sort of, finer scale microstructure simulation that we can compare to that dynamics and do sort of a computational XPCS, then we might be able to actually, look at the structure, analyze these simulations, and figure out what do each of these complex patterns mean. Before we get into the actual experiment, we'll have a better idea of maybe what's happening in the microstructure. So that would be kind of the best thing to do, because without a way to track particles, to track the structure, there's no way that we can, figure out what this crazy thing that sort of looks like a bird is meaning, right?
Yep, thanks. Any other question for Jay from the audience? And how general do you think, Jay, is this? And the techniques seem fairly general in terms of applying it to other classes of problems. Any comments on that?
So we haven't actually tested on any data other than just this colloidal suspension, but I think that you're right. The technique should be pretty general. It's all just going to be about retraining with applicable XPCS data, because the dynamics are going to be completely different system to system.
All right. Okay. All right. Any question from the audience? Very quiet audience that we have. If not, okay, let's thank Jay again for a great talk. I think that brings us to the last talk of the session. Michael, are you? All right, Michael is here. Okay. So our next speaker is Dr. Michael Service from Argonne National Lab, and he's going to actually talk to us about static and dynamic critical phenomena in rare earth separations, again using XPCS. All right, over to you, Michael.
Aha, I'm muted. Okay.
Yes.
There we go. Thanks for letting me know. Okay, great. So yeah, thanks for the introduction, Subbu. So yeah, as you mentioned, my name is Michael. I study heavy element separations here at Argonne, and I'm going to be talking about some work done in collaboration with Subbu and Brian Stevenson about separations and why we are, and kind of I'll talk about chemical separations and rare separations, then why we're interested in using static and dynamic critical phenomena to understand separation systems, and then talk about opportunities with ML and APS-U to kind of further develop this work. So let me, okay, advance the slide. Yeah, so kind of broadly, I'll start with chemical separations. We're focused on rare earths, which includes primarily the lanthanide series, which are a series of elements that are useful in a lot of technologies. One of the primary way that we're going to talk about separating rare earths or recovering them is with liquid extraction, which is a technique that is useful because it is low energy input, unlike techniques like distillation, for example. There's no input energy required. And also because it's working in liquid phases, it's very scalable, unlike something like membranes, where it's easy to just increase the volumes of the phases and get high throughput at industrial scale. So this is the workhorse technique for a lot of metals recoveries, hydrometallurgy, including rare earth separations, which is what the focus is on here. So because this separation takes place in a liquid phase, it's actually a relatively complicated system. Typically, the way that this operates is that you have metal ions that you're interested in separating, dissolved in an aqueous media. Typically, this is an acidic aqueous phase with the metal ions dissolved, and this is contacted with a nonpolar immiscible organic phase, which contains some kind of oil solvent, as well as extractant molecules, which are amphiphiles, that after you shake them together, emulsify these phases, these extractant molecules bind to the metal ions, kind of shown up here, and they pull them out of the aqueous phase and into the organic phase. And so part of why these kinds of systems are really challenging to understand is that there is quite a range of length and energetic scales present in these organic phases. They're very non-ideal solutions. We have high extractant concentrations and high metal loading, and we have interactions between these amphiphiles in the oil phase. We have hydrogen bonding as water is also co-extracted. We also have additional hydrogen bonding and protonation as we extract incidentally acid from the aqueous phase into these systems, as well as having these electrostatically driven metal extractant complexes. And so in order to do or to perform a reversible separation where we can do something like tweak the pH later and return the metal back to an aqueous phase and recycle the organic solvent, these systems are usually depend on relatively small differences in solvation free energies for the metals between the phases. And so the amount of metal that goes out into the organic phase and comes back into the aqueous phase it's a free energy driven process. And so in order to get reversible processes, we have differences in this energy kind of on the order of thermal energy. And so there's a lot of considering the complex structuring in these organic phases is necessary now to understand these relatively small differences in free energy. And so another phenomenon that's important that is related, as I'll discuss, to the structuring in these organic phases is a deleterious liquid-liquid phase transition that results as a because of the incompatibility between extracting all these polar species into a nonpolar organic phase. And so as you extract too much of this acid and too much of this metal, they become insoluble and they crash out into a dense phase, as kind of illustrated here. And condensing all the metals into this dense phase is an incompatibility with the processing equipment that's usually used when these are done at very large scale industrially. And so this practically puts a cap on how much metal we can extract at a time. If we extract too much, then it will condense out into, or not condense out, but separate out into this into this dense phase. And so we can only extract so much metal at a time. And so we're generally interested in understanding this phase transition, this liquid-liquid phase transition that occurs within this organic phase, so that we can learn how to design separation systems that let us get more metal out. And part of what makes this so complicated is it's understood kind of empirically, and it's known that basically every choice you make about the separation system has some effect on how much metal can be extracted. And so things like the molecular structures of all the species, including the solvent and the extractant, as well as the compositions of all the components in the aqueous and organic phase, and then thermodynamic properties like temperature all affect this phase transition. And so in order to kind of broadly understand the aggregation phenomena that we see in these systems and the phase transition in general, in this project here, we're talking about using the theory of critical phenomena to kind of give us a unified description that applies across many different types of separation systems, and kind of give us a way of understanding this phase transition. And so in general, in these systems, we have what's called a UCST, or upper critical solute temperature or solution temperature type phase diagram here, where at high temperature, we have a single phase. The organic phase is stable, and so there's only one organic phase. And then at certain lower temperatures below a critical point, the organic phase splits for some fraction of these extractant concentrations here. And so this is the general shape of the phase diagram. So typically we want to operate here in this single phase region where the organic phase is just a single phase. And then when we're operating under these conditions, we observe critical fluctuations here in this single phase region that stem from the critical point associated with this liquid-liquid phase transition. And so to kind of talk a little bit more quantitatively about these fluctuations that we'll be measuring with small angle X-ray scattering and then studying dynamics in these systems with XPCS, I just want to kind of discuss a bit about critical fluctuations where we have, for example, in an Ising model, we can illustrate these fluctuations here, where as we cool the system down within the single phase region and approach the critical point, we see a growth in of the correlations present in the system. And so we go from having relatively short length scales present, where the correlations decay very quickly spatially at high temperature. And as we increase or decrease the temperature, we see that we increase the length scale of these fluctuations, where they become very large as we get very close to the critical point. And then as we enter the two-phase region, the phases then again split. And so one of the really fascinating things about critical phenomena is that we have all these length scales present, this really complex behavior that comes out of really very simple interaction potentials. And so in the Ising model, we have just nearest neighbor interactions that then propagate these correlations out to very large length scales when they're close to the critical point. And so having this really simplified description of nearest neighbor interactions can give us all kinds of structuring. So it's really quite fascinating. And so in addition to just growing in these correlations, they actually have a very specific quantitative behavior, and this is called skilling relations. And so there are these power laws that define properties, like, for example, the correlation length, which is very qualitatively the size of these fluctuations that are present. They behave in a very specific quantitative way as we approach the critical temperature. And they follow this power law. We have this exponent here, which is given by what's called a universality class. And so another very important concept here is this idea of universality, where many different systems behave similarly, even though they are chemically or physically distinct, and the intermolecular interactions driving the phase separations can be very different. They all behave in the same way, even quantitatively, in the sense that they have these power laws, and the values and the exponents for these power laws is the same. And so I'm showing images here from an Ising model, and this is not just because it's a simple model to demonstrate. It actually gives us the same behavior in real physical systems. This is, of course, two-dimensional images here. And so in a real system, in a test tube in the lab, we'll be in 3 dimensions. And so the corresponding universality class would be the 3D Ising model. And so the fact that these systems, real systems, behave the same as these really simple model systems depend primarily on the dimensionality of the universality class. This tells us that a lot of the local information, such as topology, if you're on a lattice, or intermolecular interactions, if you're on a continuum, really doesn't matter. It just matters the dimensionality in terms of defining the scaling as we approach the critical point here. Okay, So X-ray scattering is a really great tool to study critical fluctuations in these systems because it gives us access to a couple of these exponents I was talking about that are then related through a scaling relation like this. And so in this particular system here, we can take a static X-ray Sachs pattern here, and the shape of the scattering when the system is dominated by these critical fluctuations is a Lorentzian function here. This is called the Ornstein-Zernicke equation. And so this gives us access, as if we study the temperature dependence, to a couple of these different exponents here. And so kind of to highlight the universality, what we do is we find, as I'll kind of explain more in these further slides, we can change a lot about the chemistry of the system. We change a lot of the parameters in the separation process, and we get the same type of scattering. We get this Lorentzian function, and all we're changing here are these two parameters, this correlation length and susceptibility parameter here, that change essentially the width and the maximum of this scattering distribution, but they don't fundamentally change the form of the scattering present. So we can look simply at binary mixtures and understand how, in simplified organic phases before we even contact them with any aqueous phase, we can see how the structuring is affected by tweaking the molecular structures of these molecules, which gives us insight into how molecular design will affect the phase behavior. So for example, here, as we kind of scan across how much extractant we have versus how much is solvent in this volume fraction unit here, we see that for some middling volume fractions of these extractants, we can grow in very large critical fluctuations when we have shorter extractant tails, these alkyl tails, or if we have larger solvent tails, basically tuning the miscibility of these components. And so this kind of structure that we observe in this single phase region can tell us about how the phase diagram is changing and how the critical temperature for these mixtures is moving based on how compatible these two molecules are. So in this particular case where we have this molecule in the upper right here, mixed with hexadecane, for example, we can get the phase boundary relatively close to this gray line representing room temperature, where it's easy to make some measurements. So if we take that sample here where we have the critical point is relatively close to room temperature and relatively accessible, we can measure these exponents I was discussing before by slowly cooling the sample down and approaching the critical point. So as we start the sample here at 25 degrees, we cool the sample down, and we see a growth in of the low Q scattering. And we see that once we compare all these different systems onto the same, collapse them onto a master curve by scaling by I0 and the correlation length, that all these temperatures are behaving qualitatively the same. We're just changing these parameters here. And then we can plot these parameters as a function of the distance from the critical point and fit the critical exponents and find that these systems are behaving like we expect, which is to say that they have the same exponents as we find in the Ising model. Okay, so that's a relatively simple binary system, so it might make sense that it behaves like the binary 3D Ising model, but we can actually consider much more complicated systems here now, where we have extraction of these lanthanides as well as co-extraction of water and acid. So now we have 5 distinct species present in these systems, or 5 distinct components that form a plethora of species. And we can compare a couple different extractants here. We have this one on the top here is interesting because it does a better job of extracting more metal before it phase splits than the one down here. And so for a given system with the critical composition near room temperature, by tweaking how much acid and metal we extract, we have somewhat more metal in this case than down here. And we also consider 2 extremes on the lanthanide series, where we have what's called a heavy lanthanide, which is later on in this series, and then we have cerium here, which is a later lanthanide. And so again, we see that we extract more lutetium. It takes more lutetium to get the phase boundary to room temperature or close to room temperature than it does cerium. And so we're able to kind of consider combinations of extractants and lanthanides. And when we get them in the same relative position to the critical point, we can take these measurements where we again cool the sample down and grow in the fluctuations in these systems, and they all collapse onto the same kind of master curve here. And again, we can then determine the scaling here as a function of temperature for both I0 and the correlation length. And we see that they all follow the same 3D Ising exponent predictions here for the scaling. And so this tells us that even when we have this very complicated system, we can define it with a scalar order parameter where we have a composition parameter here on this x-axis. So this simplified phase diagram here, where I only have one composition variable and one temperature variable, is actually useful for describing these really complicated systems, where the length scales diverge along a single composition, which is defined here on the x-axis. And then when we add more of these extracted species, really what we're doing is we're changing TC on the temperature scale, where we're changing where the critical point is. And so we can think about all the different components in these systems as kind of collapsing onto these two dimensions in terms of how they define the structuring in the organic phase. So this is, for the separations perspective, very useful because it tells us how to think about all these different components we have in the system. It's not just some hopeless case where we have way too much going on to possibly be able to make predictions about phase behavior. We can actually use critical phenomena to make a very explicit quantitative connections here. So just to kind of summarize these ideas, we have, if we just scan around in the single phase region here, we get this pattern I was discussing in the length scales that we observe in the single phase region. If we take a slice at a fixed composition, then we get the scaling that we know from the scaling relations that I discussed before. And then if we take a slice in the other direction at a given temperature, then we observe this kind of pattern here, where we see there's this ridge-like behavior in the correlation link maxima that extend out from the single phase region. So overall, we can kind of create a bigger picture here of what the structure looks like across these different systems. Okay, and so to kind of also mention how understanding, or how we are understanding the role of all these different species, is that we're performing some high throughput measurements at sector 12, where we were before the APS shut down on Monday. Some high throughput measurements at sector 12, where they have a robotic liquid handling system, where if you can tell from this image here, we have a tray on this platform that we put samples into. And we have this arm, which then goes down and pulls up a sample from the tray and moves it in front of the X-ray beam here. And we can take measurements, and this robot then returns a sample and cleans itself out. And we can essentially take much more data using this kind of a setup than we had in the past, where we had to manually load samples into capillaries, which was very slow. And so one of the main ideas that we want to understand is we want to explore this relatively high dimensional phase diagram, where we have all these components. We have how much of the metal we have the size of the lanthanide, essentially, as we go across the series, the size of the ions change. We have acid that's present in the aqueous phase that gets extracted. And so we have all the molecular structures of the extractants and the solvents. So we have all these variables to play with, and it's really hard when we're only taking on the order of tens of data points each beam time in order to get the big picture here and understand how all these different variables interrelate and play off each other. So with this kind of high throughput methodology, we're able to get the bigger picture and understand all the interplay between all these components much more so. And so we're still not automated in when we prepare the samples, in the sense that we don't have any kind of similar analogous robot to help us prepare the sample. So we're still being the robot ourselves in the lab when we make these, but at least when we go to the APS, where we're under the time crunch of the beam time, we can collect lots of data with this high throughput methodology. So I think there are some interesting opportunities here for making this further, some kind of further high throughput kind of machine learning analysis of this data. And so we can, as I'm kind of showing here, just map out phase diagrams more completely. Okay, so now I'll talk a bit about the dynamic measurements that we're taking with XPCS. And so XPCS has been talked about quite a lot today, so I don't think I need to explain too much, but we essentially take we measure one photon events with a coherent X-ray beam in time series, and that we measure this correlation functions of these photon events. And so we can construct these G2 functions and then average over some Q range here on the detector. And then for each different Q range, there is a different decay time, which is this G2 function. So we can then obtain the dynamic lifetimes as a function of Q as we measure a sample. And so for these kinds of measurements, in order to get the coherent flux that we need in order to take this data, we have to be relatively close to a critical point with relatively large length scales built in. And so in this particular case, we can cool the sample again as we're very close to the critical point and watch as we see the length scales change as a function of the Q value here. So we have this, again, this exponential relationship here where we have some Z effective exponent that gives us the relationship between the Q value and this and the lifetime that we observe. And as we cool the sample, we see a crossover in behavior from an exponent of 2, which corresponds to typical diffusion behavior, to a value of 3. And this is kind of consistent with the dynamic critical phenomena theory, where there's this crossover as we get very close to the critical point. And so in this way, we're able to use dynamic measurements to kind of study or understand the dynamics in these very complex solutions with the theory of critical phenomena once again. And so one of the things we do when we analyze the data this way is we miss out on some information. We're averaging over these Q-bins, and we're averaging over all the correlations that are present. And so in theory, there's a lot of information about how these complex liquids are moving, where there's correlations between different length scales as we have these complexes of different sizes diffusing together in solution. And so in order to understand or interpret in kind of real space what's going on in terms of the dynamics of these complex solutions, we're also developing some molecular dynamics models using machine learning in order to access the relatively large length scales that we need in order to see this critical dynamic behavior. And so from simulations, we can directly compute from trajectories, speckle patterns here, and then we can relate the speckle patterns we observe to what we see experimentally and kind of understand how the dynamics are correlated across length scales, because we have all the real space information from these simulations. And so in order to access larger length scales than are traditionally available from using atomistic simulations, which is kind of the default way of simulating liquids, is by using a technique called coarse graining, where we group together regions of atoms on these molecules into single sites, and then we create a different intermolecular potential for these beads here and train the parameters on some dense or some electronic structure calculations. And so we have this high accuracy DFT interaction energies that then we can use for a coarse-grained model that lets us access larger time and length scales. And so we're hoping that this methodology lets us overlap more significantly the types of the experimental and simulation data here. And so to kind of recap and talk a bit about the role of the APS upgrade. So from the separations perspective, we've kind of been able to simplify our understanding of all the different parameters that go into a separation system and how it affects structure and how it affects phase behavior, which is really exciting. And then in terms of making measurements and dynamic measurements in particular, we have this improvements in detector speeds, and so we're using a megahertz detector, which is very helpful. This course, okay, I guess I'll say like down in this region here at large times, or at large times and large lengths, that's easy to access experimentally. And then it's these small times and small lengths that are easy to access in simulation atomistically. And it's really kind of getting these two to overlap is the big challenge here. And so the improvements in the detectors have increased the time scale significantly. With the APS upgrade, the increase in the coherent flux will give us access to, again, smaller times and smaller length scales. And then we're moving out the boundary of the simulations using these coarse graining schemes here. And so instead of going from before, where we had essentially no overlap between the simulation and the experiment, we now have more substantial regions of overlap here. And so we're kind of excited for what we're able to be able to do with the APS upgrade. So, okay, I'd like to thank the co-PIs on this project, as well as the beamline scientists at Sector 12 and Sector 8, and then funding through BES.
Right. Thank you so much, Michael, for an excellent talk. Questions for Michael? So, Michael, where do you see like the scope for? I know you basically pointed to the modeling side where the AI ML could actually be useful, but in terms of directly like extracting information from the XPCS experiments, where do you see the scope for AI/ML, especially related to the critical phenomena that kind of is an area that many of the chemists are especially interested in.
Yeah, so I think that there is a lot of use for AI/ML and interpreting the speckle patterns, but then connecting it to the real space behavior of the complex solutions, I think is something that is, I don't see how that's possible with just the Q space data. And so I think that's really why the simulations are needed in order to make that connection. And so I think these correlations could come out of just the speckle patterns, as I think have talked about a little bit earlier today, other people have. But really it's that connection that I'm interested in that I think comes from the fact that we can do simulations and get speckle patterns from those, and then that helps us learn what different features and correlations in speckle patterns means.
Yep, thanks. Any other question from the audience? And Michael, you've also been involved in MD simulations. You've done all atom simulations yourself. In terms of multi-fidelity scale bridging, could you comment further on, where do we actually see the opportunity? I know you showed the graph where with the APS upgrade, there is a more overlap. What would
yeah. So, okay. So I think that the coarse graining is important. So this shows that we just get lots more flux, but that's actually kind of not the whole picture. When we have lots of flux, we end up with lots of potential beam damage. And so we don't necessarily get to take full advantage of the flux that we have. And so I think we want to put ourselves in the best position possible by pushing the boundary, both length scale and time scale on the simulation front. I think that's really important. Just if there's kind of this nominal overlap, it doesn't necessarily get us the whole way there.
Great.
Yeah.
Yep. Any other question for Michael?
I'll ask one. Michael, great talk. Looking at this plot and the previous slide where you showed, using simulations to interpret X-ray science data. Do you think the coarse grain models get you to the timescales that you show in the next slide as well, or is there a gap between the two?
Yeah, that's a really, really great question. So here we kind of have this power law that tells us what timescales we can access, but the dynamics are done in serial. And so there's still a large wall time associated with very large timescales, and you can't just throw more cores at it in the way that you can with increasing the length scale. And so I still think that the times is more of a limitation kind of than this plot would imply here. And so I think that it's going to take both the coarse graining as well as the flux getting us access to the smallest possible time. We will have access to microsecond simulations with the coarse graining techniques. And so I think that's kind of a reasonable objective.
Awesome, thank you.
Any other question for Michael? If not, let's actually thank Michael again for a great talk. And that brings us to the end of this particular session and the workshop for today. Join us back again tomorrow at 9 a.m. for another set of wonderful talks. So I'll see you again tomorrow at 9 a.m. Take care, everyone.
Thanks.
Bye.